Hello everyone, I'm Dr. Jack West, medical oncologist and founder and CEO of the Global Resource for Advancing Cancer Education, or GRACE. We're going to continue with the next part of our program on molecular markers in lung cancer, who to test and what to test for. I was joined in this program by Dr. Charlie Rudin at the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins University in Baltimore, Maryland. Also Dr. Alice Shaw, assistant professor and thoracic oncologist at Massachusetts General Hospital and Harvard Medical School in Boston, Massachusetts. Also Dr. David Spiegel, thoracic oncologist and director of clinical research and the lung cancer program at the Sarah Cannon Cancer Center in Nashville, Tennessee. And finally, Dr. Glen Goss, the chair of the Lung Cancer Committee of the National Cancer Institute in Canada and professor at the University of Ottawa in Ontario, Canada. So I'd like to turn, before we get into the question and answers, to Dr. Goss talking about the feasibility of doing this kind of work across a platform, potentially even national policies. And you can speak to how you've approached that.
Yes, Jack, thank you for inviting me to be a member of the panel. It's been an extremely interesting afternoon, I have to say. I've been asked to speak on the feasibility of integrating molecular markers across comprehensive healthcare systems, the intersection of benefit with cost. Now, in the medical literature, there is not a lot that has been written on this. There may be in the financial literature, but I'm not familiar with that. But in the medical literature, there hasn't been. If anybody's interested in this topic, what I would suggest you do is look at the article. It's the Lancet Cancer Commission that was published in Lancet Oncology in September of 2011, so last year. And it's a getting together of all the great minds in oncology to come up with some common themes, etc. I'm going to talk in general about some cancer care costs first, and that is, in 2008, the worldwide cost of cancer attributable to disability and premature death was about $895 billion. That's not actual care costs. That's got nothing to do with care. That's just the effect on society. And it's also estimated that the efficacy rate of drugs is about 50%. So that takes all stages into consideration and all tumor types. For instance, the last 6 weeks of life, patients often get many anti-cancer drugs that are actually of no benefit whatever to the patient. So about 50% of all drugs delivered don't do any good to the patient. This annual waste from misdiagnosis and mistreatment accounts for about $350 billion. Turning from the worldwide view to the US vision, the US federal government spent in 1990 about $27 billion on cancer care. By 2008, it was $90 billion, and by 2020, it's going to be $157 billion. So over a period of 30 years, there's been a 600% increase in the amount of money that has been invested in cancer care in the US. That's a huge amount of money and a huge increase. Opposed to that, in the private sector, in 2009, US personalized medicine market was estimated as being about $232 billion and is expected to grow annually at about 11% to $450 billion by 2015, which we know is only in 2-3 years from now. So it's not that as a society we cannot afford the current cost. We can. There's absolutely no doubt. It's just a question of setting our priorities, what we want to pay for and what we don't. But we certainly cannot afford that incremental cost on an ongoing basis. There has to be a ceiling to that. And that's where this personalized medicine and molecular profiling of patients and their tumors comes into play. This is a slide that has been shown in various forms by all the speakers to date. But I'd just like to make 3 points which have already been made in our discussions. And one is that not all of these targets are druggable at the moment. So, for instance, the biggest chunk there, which is KRAS, is not druggable, and we've discussed that. Not all of these targets are going to be important. That's the other thing. Although there are now drugs for a lot of these targets, a lot of these drugs are only available in clinical trials. So what I'm trying to do is I'm trying to temper patients' expectations by saying, look, even though we can do the molecular profiling, we don't have the answer, even if we do necessarily get your molecular profile. Although with time, increasing numbers of patients are being able to be treated or put on a trial where there would appear to be active treatment. To start with the US have been the world leaders in this molecular profiling of lung cancer and of other tumors. The two big and earliest promoters of this outside of Harvard and Baltimore have been the Lung Cancer Consortium, which Charlie has spoken about, and the Memorial Sloan Kettering, where they have been genotyping all their tumors since January of 2009, and they've looked at 7 genes in 40 mutations. Now, the important point about this is that at last year's presentation, when Mark Chris presented the Lung Cancer Consortium data, he said that only 13% of patients were actually found, an active treatment was found for them, despite the fact that they were profiled. So I want you to remember that number of 13%, because it's going to come up again later in my talk. This snapshot, this is one of the multiplex profiling, and I'll just put this up just to inform people that you can detect these mutations in a very small amount of DNA, so as little as 5% of DNA. So we don't need a lot of tissue, actually, to do this molecular profiling, but we do need tissue, as has been clearly made earlier on. When you speak to medical oncologists in academic meetings, they will tell you that we can profile 100 tumors for $200, all right? And if you speak to scientists, they'd say, oh, well, the price is coming down, and we are soon going to be able to do genotyping on thousands of genes at $50. The price is coming down. So what I did was I got my research assistant to phone a few places and asked her to try and price this in real terms. So I'm not picking on Vanderbilt here at all, because this is probably true of most of the places where they provide this technology. So at Vanderbilt, this is what you get with Snapshot. These are the genes. There are 8 genes, and those are the mutations that they look for as listed on the slide. But this is the cost. This is the actual cost. So if you're a patient at Vanderbilt and your tumor is going to be genotyped with Snapshot, it costs you $1,400. Vanderbilt will do the same genotyping on somebody who's not at their center for $1,700. Now, if you batch them, so if you're a physician who has multiple lung cancer patients and you can batch them, and I think this scenario is remote, is not common, you can get it for $1,600 for 5 patients if you're at the VU. But if you're outside of the VU, it costs you $2,000. This gives you an idea of the actual cost to the patients. It's somewhere around $1,500 to $2,000 to have your tumor type for 8 genes. So that's about the cost. Well, what about in other jurisdictions outside of the US? What are they doing? In the UK, in 2011, which is approximately 2 years after the US started their endeavor, so in 2011, the UK's Technology Strategy Board and 2 big pharma companies have funded an exercise where they are going to genotype 9,000 patients in 7 medical centers. These patients are going to be genotyped for free. This incorporates all tumors, and these are the mutations that they're going to be looking at. And as you can see, that most of those mutations, there are actual drugs in particular cancers for those mutations. So if you have wild-type KRAS, then in colorectal cancer, there's appropriate treatment for you. If you have an EGFR mutation, non-small cell lung cancer. So they've chosen pretty conservatively and quite carefully that panel of genes. In France, a network of 28 hospitals linked across France, the National Cancer Institute and the Ministry of Health have funded a genotyping exercise which involves the genes listed there. And it costs about $9 million a year of public money. And they believe that they're going to save many, many more millions of dollars if they do this. In Canada, led by the Ontario Institute of Cancer Research and the Princess Margaret Hospital, 5 cancer centers, including my own, have done a feasibility study where we've looked at 100 patients. And what we've done is we've compared 2 systems. So we've compared the Sequenome system with the PAX Bio system and looked for validation across both systems. And what we have screened for is 238 mutations in 19 oncogenes. And one of the feasibility metrics was to be able to deliver a result in 21 days. So that has been accomplished. On the initial 15 patients, there were actionable mutations in 57% of patients. That was pure serendipity, because that rate has fallen to 12%, which is exactly the same as Mark Chris's 13% in the Lung Cancer Consortium. So actionable mutations, somewhere between 10 and 15% currently. Now, that is obviously going up in places like MGH. I'm sure it's higher than that by now. But that in the general public is about what you get. As a result of this, we're now going to enroll 500 patients on another study looking at 25 oncogenes and genotyping them. And on this occasion, we want to get the molecular profiling data into the electronic medical record. This is an issue in Ontario because of the legislation, and that's why this study is so important. It's got to do with privacy legislations and this whole area around genotyping and the legislation around it. So this is a pilot study to see if we can deliver that. This is my final slide. What are the advantages of molecular profiling or biomarker-driven cancer treatment? Well, the first is we avoid time wasted on drugs that are not effective, and patients are not subjected to the toxicities of non-effective treatment. Secondly, in general, the new generation of targeted agents have fewer side effects than our older chemotherapy. So you are saving toxicity, you're delivering more effective treatment, and you're not creating toxicity in patients that are not going to benefit. This improves health outcomes, and because of the size of clinical trials, and this has been alluded to both by David and a number of you, the cost of clinical trials is hopefully going to fall. The additional spending, so this is the theory, that the additional spending on innovative targeted medicines will be offset by savings made from more effective and tailored strategies. So that's on the plus side. On the minus side, or the disadvantages, are that, number one, we have not had a lot of validation. So a lot of the biomarkers that are being tested for and patients being put on treatment for have not been properly validated. Also, the regulatory agencies have not come out clearly on what they want. They've come out on individual biomarkers, and they've written quite extensively on what they consider are minimum standards for biomarkers. But they haven't addressed this issue that we are all subjected to on a daily basis, and that is multiple biomarkers within a single screening profile. So then we need some guidance on that. And in lung cancer, there's nearly complete absence of evidence from randomized controlled phase 3 trials supporting the utility of a particular marker. To date, as far as I'm aware, there's no trial that has randomized patients based on a marker rather than on treatment. The other issue that we need to address is the issue that has been alluded to by all the speakers, and that is the analytical validity of the test that we are doing. So what I mean by that is you can send the same lung cancer to one institution, and that institution will report the tumor as being EGFR positive, and you can send that tumor to another institution and the patient will be reported as EGFR negative. Now, this cross-validation, and not a lot of it has been done, but when you do it, and I've been involved in 2 studies where this has been done, you find this incompatibility of results. So we need the test to be validated, analytically validated, and to be robust. And then using those tests, we need to design the validation clinical trials. And that's the way we have to move forward.
Well, thank you. I want to pull back and ask a general question. You brought up a big issue, and that's cost. We're hearing more about it. We didn't used to think about it. And the general rule about oncology was that it's inelastic, that we don't really factor in costs. We can't because the stakes are so high. It's becoming harder to not think about it, and it's being published more about, and it's being talked more about, both in the general press, in the lay press, and in our own meetings. The question is, these tests have costs, and the drugs are costing more and more. As clinicians, is it our role to even think about this, or must we divorce ourselves from that entire process and say we have to do what's best for our patients and not factor in cost to society? It's a hard one.
Well, none of us like talking about cost with the patient when it comes to trying to help them. I think the best argument, one of the best arguments that can be made for all of this that we're doing from a cost perspective is, and Charlie alluded to this with that New England Journal IPASS study, is avoiding what could be an expensive therapy in someone that is not going to benefit from it. So we'll pick on Tarceva for a moment. So a drug that can cost anywhere from $1,500 to $3,000 a month for a patient or an insurer, not giving them that drug when they don't have the target that drug was designed to inhibit. So there are cost savings there. And I do believe what Glenn was saying in that there are all these downstream savings that hopefully can occur because you're not giving patients carbotaxol and gem and taxotere because maybe they're staying on a drug like Xalkori for a year and a half. Now, you can make an argument, aren't you just deferring or delaying those costs because those same patients are going to get carbotaxol and Alimta or whatever down the road. But my sense is probably the duration on those therapies won't be the same as if they got it first line. But cost is always a nightmare, and as technologies advance, their company X, Y, or Z has a new panel that can be done for a little bit less or a little bit more, and who's going to pay for that? I just recently sent Alice a sample from a patient where it was for Ross testing. Alice has one of the few centers in the country that offers it, but Glenn brings up the point, what are you going to do if that result's positive anyway? So this was a long discussion with the patient. The test, not to pick on Mass General, I thought it was a very reasonable cost, was something that was much lower than that. It was actually a few $100s, $600. I had a discussion with the patient about the small chance that would be positive, and if it were positive, I don't know that I could get her access to Xalkori, but maybe I could steer her towards a clinical trial that had a drug, a next generation ALK inhibitor. And with that discussion, she was willing to pay out of pocket that cost to have that testing done. And similar conversations I've had with patients, they've been willing to do that, but most patients cannot afford this expensive testing. So I don't know that we have the answers on that.
In the ALK world in particular, it's a big issue because unlike the mutation testing that we've talked about, EGFR and KRAS and PI3 kinase, where you can kind of batch it all and do it sort of in a multiplex way, ALK and ROS and RET, there are all these individual FISH tests. And the way Xalkori is approved, of course, is in conjunction with the diagnostic that's approved by the FDA. And so depending on the insurer, they can insist that FDA-approved kit be used. Now, the estimated cost for doing that type of testing then comes out to anywhere between $1,000 to $1,500 per patient. So if you think about an ALK patient and the frequency being, say, 5% in our population, that means we have to screen 20 patients or pay perhaps $20,000, $30,000 to identify one ALK positive patient. And that cost has come up many times when we've met with other oncologists, especially from out of this country, who think about those costs and just think there's no way they or us actually can really manage costs like that. Like you were pointing out, we don't screen most of the patients in this country. And if we did, and we had to pay $20,000 to find one ALK positive patient, it would be formidable.
Yeah, these are issues that we are having to wrestle with. In addition to wrestling with just the cancer, we now have the financial thorniness of it.
So to try and answer your original question, I think as physicians, our primary responsibility always is to our patients. But on the other hand, society can't do this without physician help and input. So we have a responsibility to our societies to provide some leadership and some advice on how best to move forward. And so in our individual practices, we have to do what's best for our patients. As societal animals, we have to try and find a way to deliver the best possible care at the most reasonable cost. I think that is a societal responsibility that we have.
People had some questions, and my apologies to the online audience that we will not be able to get to everyone's questions. So one is the issue of reflex testing in early stage patients. The NCCN guidelines are for advanced disease, and that's where we have it tested. Do you recommend mutation testing in earlier stage patients, and how would you use that information now? I'll open up to anybody who would speak to it.
So I can certainly speak to what we do. We're not doing it, and we're not pursuing it because we would not use that information to inform treatment decision. As has been pointed out, EGFR mutation in that context, it's not so clear that they should get Tarceva. And in fact, for those patients who are appropriate for standard chemotherapy in the post-surgical setting, we would recommend chemotherapy regardless of the mutational status. So we're actually not doing that. Having said that, if and when those patients recur, we certainly want to know their mutational status. So we often go back and look at those in retrospect, but we're not doing it reflexively except in advanced stage disease patients, at least at our institution.
Other comments?
Yeah, I think that's the right answer. You're aware of the CAP-IASLC guidelines that are supposed to address this issue. It's supposed to come out any day now. They had a public comment period in the last month, and they're going to address this very issue. So we have reflex testing at our center, but trying to limit that to patients with only advanced disease. I think ideally, if a patient, when they recur, you want a new biopsy anyway, probably, to see if the tumor is actually maybe has a different genotype than what they were diagnosed with.
I don't think there's anything wrong testing this conceptually in the form of a clinical trial. That's fine. But I'm in agreement with Charlie and with David that in the adjuvant setting, there's no role for it at the moment.
Well, I think that part of the problem is once you have that information, it sometimes leads you to bad decisions. That's my concern about it. Final question, and you alluded to this. If somebody recurs, you're going to want tissue. In fact, I think we could probably all agree that it would be most valuable to have the tissue at the time of recurrence and not just rely on their surgical biopsy material from 2 years ago anyway, because of the sense that tumors change over time. That leads to the question of serial biopsies, which we haven't talked much about. We were really talking, the strongest toehold we really have on this is early at the time of first diagnosis. I know that at Mass General, you are routinely at least trying to get biopsies. Can people speak to your perceptions now about risk versus benefit of repeat biopsies, and what do you actually do with that information if you have it?
So at our institution, we do routinely perform repeat biopsies, but primarily in the context of acquired resistance to Tarceva and Xalkori. For example, of course, if we have someone who's relapsed, we do go back to the surgical specimen unless it's exhausted or not available. But in the context of acquired resistance, we definitely want to rebiopsy people, and we direct patients to clinical trials based on those results.
I think it comes down to either you have a trial that's looking at rebiopsy, you're looking for resistance patterns. But really, are you going to do something with that information? I'm fortunate to be at a center that has a very large phase 1 program. So it's easier for me to have a conversation about how that could lead us to steering towards one of 70 clinical trials that might make more sense than just picking one out of a hat. But I think if a patient doesn't have ready access to act on that information, or the doctor, I'm not sure how helpful it will be right now. I mean, I think it's ideal, but I'm not sure from other than an academic standpoint, it would help patients who don't have access to clinical trials.
I think Glenn made a very important point earlier about the relative perspective of this from patient perspective versus physician. As doctors, we're taught, first do no harm. Really don't do things if it's going to be potentially dangerous unless it's really going to influence your care. But I think patients who have advanced lung cancer are willing to take risks that are going to be potentially informative for them in terms of guiding future therapies. And I think increasingly, at centers like up in Boston, certainly, and the other great example of this, I think, is the battle trial that was done at MD Anderson. Patients are willing to do this, and I think we need to listen to our patients.
That I agree with. I think that makes sense. We haven't really talked about actual risk of the biopsies, and I think our perception is that it's pretty minimal. Occasionally, people will have a pneumothorax and need to be hospitalized with a chest tube, but that tends to be the extent of it. It's not zero. And the other rule that we have right after above all do no harm as the first rule of medicine is, a lot of us are taught, if it's not going to change your management, don't do a test. Yeah. And I think that's right now the thing we struggle with. How much do we invest in this could be useful in the future, even if you can't use it now? That's, I think, a challenge.
And hopefully in the future, these newer technologies like circulating tumor cells and circulating DNA will be developed to a degree where we have confidence in them and can base our testing on.
That would be amazing.
And then patients will not have to be biopsied.
Right. Well, I want to thank our panelists, and I want to thank the Longevity Foundation for our partnership with them that made this program possible. So thanks very much, and have a good night, everyone. Thanks for listening. If you like and learn from our GRACEcast, you can subscribe on iTunes by just searching for the term Cancer Grace, find podcasts in the subject you want, pick a format of audio or video, and then just click subscribe. It's that easy. And for those of you who don't want to miss any of our programs, there's even a feed for all subjects. You can also find us on YouTube at Grace for Cancer Info, and that's the number 4 in one word, Grace for Cancer Info. Finally, if you haven't been there yet, please check out our GRACE website at www.cancergrace.org. And don't forget that donate button in the upper right. Our content, which helps tens of thousands of cancer patients around the world every month, is made possible by your support. Thanks again.